So hello everyone. In this video, we'll study about the subcellular fractionation. Okay. In previous video, we have finished the structure and function of prokaryotic cells, right? And in prokaryotic cell, there are no sub organelles. There are no cellular organelles. Okay. So we have there is no need of study the organelles because there is no organelles present in prokaryotic cells. Okay, cell organelles are only present in eukaryotic cell, right? So, with the help of electron microscope, we can visualize only the cell structure, only the structure of the cell we can know. But the this is not sufficient for knowing the function of the various components of eukaryotic cell. Okay, there are various components, various organelles that are present in the eukaryotic cells. We know that is endoplasmic reticulum, that is lysosome, peroxisome. Okay, there are many organelles or sub organelles are present in eukaryotic cells. Okay, so we can know the functions only after the isolation of the subcellular organelles, right? So then isolation of cell organelle is required. Okay, isolation of the cell organelles is required to know the function of the particular suborganelles, and this process can be done by but the process that is differential centrifugation. Okay, a method developed by Albert Claude, Christian Didouve, and their colleagues, right, in 1940s and 1950s. Okay, to separate the components of cell based on their size and density. Okay, the differential centrifugation is the process, okay, that can separate the components of cell based on their size and density. Got it? Then steps of fractionation. Okay. Before fractionation, we should know the protocols of cellular fractionation, right? So the first step in subcellular fractionation is the disruption of the plasma membrane. Okay, that is the disruption. The disruption means breakdown. Okay, breakdown of the plasma membrane. After the disruption of plasma membrane, we can isolate the cell organelles or subcellular organelles, right? Plasma membrane under a controlled condition. Okay, what is controlled condition? The condition in which the we should know the we should apply the method, that is that we should apply the limited chemicals also that can only disrupt the plasma membrane. Okay, or if this chemical or if this method will fractionate or will disrupt the organelles, then how can we isolate and how can we study the function of it, right? So that is the control condition, only the disruption of the plasma membrane, not the disruption of organelles, that is endoplasmic reticulum, peroxisome, lysosome, mitochondria, chloroplast. Okay, so after that, all the procedures can break the plasma membrane and endoplasmic reticulum into smaller fragments. Okay, smaller endoplasmic reticulum into smaller fragments. Okay, that after the into small that after the endoplasmic reticulum break into smaller fragments, we can isolate only. Okay, then other components of this cell, that is nuclei, lysosome, peroxisome, mitochondria, and chloroplasts, intact. Okay. Then methods for disruption of plasma membrane. Okay. First, we break the plasma membrane. We broke the plasma membrane under a control condition. After that, all the procedures can break the plasma membrane and endoplasmic reticulum. Got it? Then other components. Okay, the control condition, that is, I have mentioned that under which condition that do not destroy the internal components of the cell. Okay, that do not destroy the internal components, that is the mitochondria, chloroplast, subcellular organelles that can't destroy. Okay, then other components of the cell, so nuclei, lysosome, peroxisome, mitochondria and chloroplast intact, remain intact. Okay, then the methods for disruption of plasma membrane. First, we sonicate and then grinding in a mechanical homogenizer. Okay, so what is sonication? That is ultrasonic vibration to fragment the cells, macromolecules, and membrane. Okay, for the disruption of plasma membrane, we will apply the sonication method, right? Then mechanical homogenizer. For homogenizing the what? Homogenizing the various types of material uh, present in the solution, right? Then a homogenizer is a piece of laboratory or industrial equipment used for the homogenization of the various type of material such as tissue, plant, food, soil and many others. Okay, then this is the probe sonicator, that is the homogenizer. See here, this is the homogenizer. Okay, and this is the solution in the beaker. This is the beaker and this is the solution. Okay, this machine can homogenize the materials. Okay, this rotating, see what this can homogenize the materials or the tissues present in the solution in the beaker, right? And this is the sonicator for creating the vibrations. Okay, vibrations from here, from everywhere, the vibration will be created and the plasma membrane will be disrupted. Got it? Then see, after the tissue homogenization, first we apply the sonication. Okay, then homogenization. Okay, this homogenization or this homogenate, this here, this is the homogenate tissue, homogenate. Okay, the all the subcellular organelles present in the solution and the plasma membrane got disrupted, right? Then we applied the low speed centrifugation. Centrifugation is the machine. Okay, the we can maintain the speed also, low speed, high speed, that is 1000. Here we have applied the 1000 g. Got it? First we'll know about the rpm and rcf, that is a g. Okay, here we can apply G, that is RCF. Okay, first of all, what is RPM? RPM is the evolution per minute, and RCF relative centrifugal force or G force. We can say that is the G force or the RCF. Okay, relative centrifugal force. Got it? Then RPM indicates the speed at which the rotor is rotating. Okay, rpm is saying about the speed, and rcf is the term used to describe the amount of accelerative force applied a sample in a centrifuge. Okay, that is the force. Okay, force that is applied to the solution, and there's rpm is the speed at which the rotor is rotating. Okay, that is a rotor speed of the rotor. Okay, and rcf is the force applied to a sample, direct sample. Okay, so here we have first from the low speed to high speed. Okay, low to high speed. First of all, we have applied the 1000 G, that is 1000 G force, that is RCF. Okay, then we have collected the supernatant subjected to the medium speed. Okay, you know about the supernatant and the pellet. Here is the test tube. This is the solution. Okay, this is the solution. After centrifugation, the after after the centrifugation process, here some amount of, here, okay, got it, collected some amount of, what, some amount of remnant is collected at the bottom. Okay, this is called pellet, and this is a solution. Okay, this is the supernatant. Okay, this is the, what, supernatant. At the bottom, collected remnant is pellet, and the solution got the other remaining solution at the supernatant. Got it? So after low speed centrifugation, 1000 G per 10 minutes, we got what? Pellet contains whole cells, that is nuclei, cytoskeleton, and plasma membranes. Got it? Then after we have collected the supernatant into another test tube. Okay, in one test tube, pellet is present. Okay, and we then collected the supernatant into another test tube. Here only supernatant is present. Okay, again we have applied the, what, high speed centrifugation, that is 80,000 G per 1 hour. Okay, then after the 20,000, first 1,000, okay, 1,000 we got pellet con wholesale nuclei, cytoskeleton, plasma membrane, all of these organelles. Then the then we have applied the high speed centrifugation, that is 80,000. Okay, the first we have applied what, 1,000 G per 10 minute. What we got? The pellet contains whole cell nuclei, cytoskeleton, plasma membrane. Then we have applied 20,000 G for 20 minutes. What we have got? The pellet contains mitochondria, lysosome, peroxisome. Okay, okay. Then supernatant subjected to high speed centrifugation at 80,000 for 1 hour. Okay. Then what we got? Pellet contains microsomes, fragments of endoplasmic reticulum, small vesicles. Got it? Then we have applied 150,000 G for 3 hours. Okay. Then we got pellet contains ribosomes, large macromolecules. Got it? First this is the homogenate. All cellular organelles are present in homogenate. Okay, then we have applied what, the low speed centrifugation, that is 1000 Z for 10 minutes. Okay, what we have got? Pellet contains whole cell nuclei. Pellet got whole cell nuclei, cytoskeleton, plasma membrane. Got it? Then the supernatant present here, we will collect the supernatant in another test tube. Again we centrifuged. Okay, this is 20,000. Again we centrifuged it, and we got what? Mitochondria, lysosome, and peroxisome. This is the pellet. Again we transfer the supernatant. What we got? Microsomes, fragments of ear, and small vesicles. Okay, here, pellet. Again we centrifuged it. What we have got? Pellet contains ribosome, large macromolecules. Okay, finish. This is the summary of the centrifugation. Understood? Okay. Then this is another diagram of what? This is the another diagram of centrifugation. First of all, suspension of broken cells containing subcellular components. Subcellular component, that is, here we have called it the tissue homogeneate. This is the tissue homogeneate, and in other word, here it is a subcellular component, that is suspension of broken cells. We centrifuged 800 G for 10 minutes. It will vary, okay, it will vary according to the book, according to the authors, okay. Here we have, here we saw that 10,000, well, 1000 G 10 minute, and here 800 G. Mm, nearly equals to, okay, nearly equals to 1000, yes or no? Then centrifuged it, we have got nuclei sediment. Sediment, that is nuclei pellet. Here it is the pellet. See, wait, it is the pellet, and here is the supernatant, okay. Here is the supernatant, and here is the supernatant. It is the cytosol. No, there are no, after the whole process there are no subcellular organelles remain, okay. Only remain is the cytosol. This is the cytosol, got it? See, then we centrifuge 800 G. Okay, then we got nuclear sediment. Then we centrifuge supernatant at 15,000 G for 10 minutes. Okay, then we have got mitochondria, lysosome, peroxisome, sediment. Peroxisome sediment, that is pellet. Here, this is the pellet. Then we have applied 100,000 per 60 minute. 1 lakh G for 60 minutes. That we have got the fragments of plasma membrane and endoplasmic reticulum sediment. This is the, here this is the pellet. Got it? Then we have then we centrifuge at 200,000 G, that is 2 lakh G for 3 hours. Okay, and then we have, lastly we have got the ribosome sediment, and here ribosome sediment are large macromolecules. Got it? Then in supernatant, in there are no cell organelles present. After the whole centrifugation process from lower to higher RCF, lower to higher RCF or a G force. Got it? And this is this supernatant, we got only the cytosol, the liquid substance present in the cell. Then here it is the ribosome sediment. Okay, got it? So we have finished with what? Finished with the subcellular fractionation technique. Got it? And in next class we will do some questions about subcellular fractionation. Okay, this is important for your gate and net exam. In plus two council exam, this is not important, but if you will appear the net examination and gate examination and any other examination for your PhD admission or MSc CPET exams, okay, this will be more important topics, the more important chapter, that is the subcellular fractionation technique, and you will get some questions from this, that is the differential centrifugation method, who developed this technique, and then we'll we'll got some questions from about the sonication mechanical homogenizer, and here this is the important, this will be the questions from your net and gate, that is we got pellet contains nucleus, cytoskeleton, plasma membrane. What in which G we have applied? Okay, and for what minute and for what time and for what g we have applied to get the nucleus, cytoskeleton, plasma membranes. Okay, then this diagram is very very important for solving the questions. Okay. Okay, then thank you.